First, watch the short embedded video. It's only about 3 minutes. Take your time, pause it, play it back, watch it until you can watch it straight through and understand protein synthesis as they explain it. After the video, there will be a step-by-step guide to fill out your graphic organizer.
Every activity carried out by living cells uses one or more proteins. How are proteins made? The process called protein synthesis begins in the nucleus of a cell within a chromosome made of DNA, which stores the genes or recipe cells use to make proteins. The process begins when the section of DNA that contains the plans for a protein is copied or transcribed. The two strands of DNA separate, and free nucleotides pair with their complementary DNA bases. The new molecule formed is called RNA, which contains the base uracil, U, rather than the thymine, T, of DNA. The RNA is set free, and the DNA ladder zips up again. The RNA strand is called messenger RNA, or mRNA for short, because it carries the genetic message out from the nucleus to a cell structure called a ribosome, where the protein is built. The average cell of a mammal contains some 10 million ribosomes. The messenger RNA joins to a ribosome. The bases along the mRNA are grouped into sets of 3, called codons. Each codon matches a complementary set of bases, called an anticodon. On another kind of RNA, transfer, or tRNA. Each tRNA also carries one of 20 amino acids. Here, the tRNA carries the anticodon UAC with the amino acid methionine. The ribosomes assemble the amino acids together into a chain called a polypeptide in the order that forms a particular protein. As the first tRNA floats away from the ribosome, it leaves behind its amino acid. The ribosome moves along the messenger RNA. Each transfer RNA brings along its specific amino acid. The amino acids are assembled one at a time in the right order. Protein synthesis ends when a special stop codon is reached. The string of amino acids has become a polypeptide chain. The ribosome then releases the mRNA and the polypeptide. The polypeptide chain folds into a three-dimensional shape that is unique to each protein's structure and function.
Okay, let's orient ourselves in the first image. Here we have the cell membrane and the nucleus. Inside the nucleus we have a section of DNA with 3 base triplets. Filling out the box to the right, we'll see DNA is in the nucleus. As we look at the second image, let's label this part of protein synthesis. It's the first step in protein synthesis. That's right, transcription. Let's follow the bullet points to figure out what should happen in the second image. Number 1, DNA unzips. Let's follow our same DNA strand from the first image. Unzip. What's going to make a copy of one strand of DNA? That's right, M, or messenger RNA. The mRNA code will build across from the first DNA code. T will get matched up with A will get matched up with, this is mRNA, so it will be a U. Pause the video and see if you can make the match on your own. The final bullet point says DNA closes back up and mRNA can leave the nucleus. In the previous image, remember that we said mRNA would leave the nucleus. Well, where was it going? mRNA leaves the nucleus and moves out to the, that's right, ribosome. The ribosome is where the mRNA will code to make a protein. Note the labeling of a section of 3 letters of mRNA. It's now a codon versus DNA where it was a triplet, base triplet. At the next image, let's stop and label that this is starting the second step of protein synthesis, or, that's right, translation. This makes sense because the ribosome will be translating the code of mRNA to bring in the corresponding anticodon. The ribosome will use tRNA to bring the amino acids to the mRNA. The ribosome reads the first code here, GCA, and brings in the tRNA anticodon CGU, which will have an amino acid attached to it. The end result will be a long chain of amino acids, which will become the protein. The next image shows a continuation of translation. The ribosome has already read the code for the first codon, and the anticodon has brought in one amino acid. Notice that the anticodon is no longer here. After it drops off its amino acid, it's free to go. The next codon that will be read by the ribosome is UGC. See if you can figure out on your own what tRNA anticodon will come in to match that. ACG, which will bring with it another amino acid. The first tRNA is left, the first amino acid is left behind and connected to the second amino acid. Okay, on to our last image and our last step of translation. Notice that throughout all of the images, I continue to write DNA inside the nucleus. You should continue to do that as well, as it's good practice of which base pairs match together. Now over at the ribosome has translated or decoded 2 codons of mRNA and only has one left to do. The last codon, AUC, will bring in the transfer RNA code of UAG. What will come with that? That's right, another amino acid. Let's follow along to the box to the right. The third tRNA meets up with the mRNA. The third amino acid is dropped off. A chain of amino acids has been formed. The chain of amino acids is now a protein. Oh, like protein synthesis? Blank are the building blocks of all cells. Proteins, of course. One other way to say this is there are many different proteins. Each protein is made up of a specific number and order of amino acids. So we synthesized or made a protein in protein synthesis? Oh, that makes sense.